We're ending our semester by looking at the diversity of plant life. And there's a good reason for this. What I found is that if you have more experience with plants, you have a better foundational understanding of plant cells, plant tissues, plant organs. You've experienced the plants that we like to eat. You have more knowledge. Then when we start to talk about why plants look the way that they do, why they have the different adaptations, why we see such a diversity of plant life on this planet, it sticks a little bit better and it makes a little bit more sense. You have to have some experience with plants before we really get into the details of the diversity of plant life on our planet. So what I've done is I've combined chapters 8 and 9, the diversity of plant life and the role of evolution, because we wouldn't end up with the diversity that we have if we didn't have the concept of evolution and those changes in a line of descent over time. So throughout the semester, I've been introducing you to phylogenetic trees. So we've been talking about the relatedness between different species, and hopefully dramatic examples come to mind. So when we think about things like potatoes and tomatoes, and the idea that those are part of the nightshade family, which means that they have the shared characteristic of producing toxins, and yet through artificial selection, through humans selecting for specific traits, we made those plants into the delicious things that we love to eat today. So throughout the semester, we've been seeing a lot of these different concepts. So again, hopefully this last set of chapters here has combined diversity and evolution, makes a lot of sense. So at this point on the planet, we have about 8.7 million named separate species. Most of those species are going to be the cute, the fuzzy. We have yet to discover a majority of the plants even. And then, of course, this squeamish, ooly-gooly stuff like bacteria, nematodes, insects. There's still a lot of those that need to be discovered and named. But even with all of the wonderful things that we have found and named, we have to have an organized way to discuss them. This is where taxonomy comes into play, the discipline of naming things. So remember, our three big domains are bacteria, archaea, and eukarya. We break the domain eukarya into those smaller kingdoms, plants, animal, fungi, and there are other kingdoms within the domain eukarya. Of course, we've been focusing mostly on plants and touched a little bit on humans, and we'll touch just briefly in this unit on fungi. Now, throughout the semester, I've been making you give me the scientific name. And the scientific name is based off of the genus and the species. This is our more detailed way to communicate about organisms. Essentially, when we use the genus species name, we are alluding to or communicating those shared characteristics that a specific individual organism that we're looking at has in common with that larger family, or maybe going all the way to the larger domain. So here is a picture from your textbook where we can see that breakdown. When we're at the kingdom level, we have a lot of individual organisms, probably not a whole lot of shared characteristics. But by the time we get to the genus level, we're looking for organisms that have a lot of similar shared characteristics. And here the example is the lily family. Now remember, the lily family also includes things like onions and garlic. And so while we see these flowers, these beautiful, luscious lilies like we dissected in the flower lab, they tend to have the three sepals and three petals, and the sepals take on colors and look like the petals. We have a lot of shared characteristics in the lily family. We also have to remember that some of those members in the lily family will look like things like onions and garlic, storing all of the energy in modified leaves in the bulb. So that's why it's important to talk about genus and species. We want to allude to shared characteristics, and we want to be able to communicate specifics about individuals. So this Linnaean system of classifying living things, of talking about living things, is exactly what helps scientists to get to the important stuff. If we can have a quick, concise way of communicating, then we can talk about the important details and why individual species, individual organisms are so important. Some necessary vocabulary that we need when we start to talk about the evolution and diversity of plant life include things like population. When we talk about a population, we've got a group of individuals from the same species. So there should be a lot of genetic similarity. They'll live in approximately the same geographic location at the same time. Speciation is when we have the splitting of one species into two or more species. And the important thing here is that we're going to get differences. Now, whether the differences look very dramatic or whether they look very similar to one another, the important thing is that we have changes in the DNA. The new species can no longer interbreed with that original species. So two other terms that are going to be important to us, especially when we look at the evolution of plants. One term will be this term ancestral characteristic. This is some kind of trait or structure or behavior which sticks around even when we have the splitting of species. So we're going to be looking for those things, those adaptations that continue even though we have speciation occurring. And then we're going to be looking for the derived characteristic. What is that structure or behavior or trait? What is that adaptation that our new species has? Does it give our new species some sort of advantage over the older species? Is there some kind of environmental pressure driving that derived characteristic, the development of that derived characteristic? So we look for these two things when we're looking at the relatedness between plants. Now, evolution is simply defined as changes in a line of descent over time. So we are looking for changes in the DNA. We're looking for ancestral and derived characteristics. And we have to keep in mind this concept of time. It's species specific. So for something like a redwood tree that can live for a couple thousand years, it may take a long time for us to see changes in that line of descent. But something that's really fast-lived, like a fast plant, we may start to see changes in a line of descent by the end of one calendar year. Remember, we could have gotten 2 or 3 generations of fast plants to grow just within the 16 weeks of our semester, depending on how quickly your fast plants were growing. Now, the theory of evolution is mostly credited to Darwin, and I find that I'm sorry about that. I wish that we had a broader understanding of all of the people who contributed to the theory of evolution. It was more than just Darwin's idea. It was so many other people who were contributing to essentially this theory unifying many different observations and trying to explain all of the weird diversity of life on this planet, why certain animals behave a certain way and why certain plants have poisons or toxins or produce chemicals that make us hallucinate or whatever it may be. People have been trying to explain this diversity of life on this planet for a very long period of time. So we got to understand a little bit more about Charles Darwin himself to understand why he gets credited with the theory of evolution. So a couple of interesting details. First, remember Darwin lived in England, and he lived in England during the time when we really had the Industrial Revolution exploding. Colonization was very strong, and people in Europe were benefiting from the resources gained through colonization and that disproportionate economic system. And so we have a very strict social hierarchy in Europe and specifically in England. If you were in a lower class, the possibility of ever being able to work out of that lower class, well, it simply wasn't going to occur. So Darwin's father was a medical doctor, so he had a fairly stable place in the English social hierarchy. He had wanted his sons to become doctors as well. This way they would have a stable future. Parents have always been the same. They want their children to have at least as good or a better future than what they themselves have. So Darwin's father put a lot of pressure on Charles Darwin to go to college. And unfortunately, when he went to medical school at the first surgery, he didn't do so well. He can't stand the sight of blood. He was skipping classes. He was hiking around in the woods, making observations, collecting beetles. He ends up having to drop out of medical school. So his father's kind of freaking out at this point because it's England. If he can't maintain a social hierarchy, he's going to have a rough future. So his father is talking to his Darwin's uncle, and then also ends up talking to a mentor of Darwin, a professor by the name, a botany professor by the name of Henslow. And between Darwin's uncle and the mentor Henslow, they convince his father that he's going to be okay. Chucky D is going to be fine. We just need to get him focused here. So what they end up doing is they contact the U.S. Naval group, and they say, could Charles join one of the voyages? At this point, the Navy vessels, a lot of them were going around the planet, and they were finding more resources to send back home to England. So could Charles join on one of those? They get him paired up with the Beagle, the ship, and the captain of that ship was Fitzroy. Now, being the captain of a ship meant that you were at a higher status in the English social hierarchy. This ends up being kind of important for understanding Darwin's role in the Beagle's 5-year voyage, finding resources to send back to England. So Fitzroy is the captain of the ship, even though he was going to be gone for a long period of time in places of the planet that didn't have the same luxuries, didn't have the same social status. He was still expected to maintain the social hierarchy. He was expected to maintain those British standards. Now, if you've never been around anybody from Britain, I can tell you that they do have this very profound sense, even today, of what the social standards are. My advising professor was from England, and he was so picky about how to make tea. It was very, very particular. Another British professor down the hall from me, he once fired one of his graduate students, kicked him out because he could not make the tea appropriately. So even to this day, we have this sort of British sense of what the social standards should be and that they will be maintained at any cost. So Fitzroy was under this kind of pressure. On the Beagle's trip, there was already a naturalist on board. The naturalist was a coveted position in European society. It was often funded by wealthy people, and the naturalists could go and make observations, collect samples. They were what would now be considered a research scientist. So there was already a person on board who had that role. And when Darwin was brought to join the Beagle journey, the naturalist got very jealous, felt very threatened by Darwin, thought that his role was going to be taken. But Darwin's role in the Beagle was to accompany Fitzroy, to help Fitzroy to maintain this social standard, to have intelligent conversations when they sat down for tea at the appropriate time every day. I mean, that was Darwin's role. So what would happen is Darwin, when the Beagle would drop off at port, Darwin would be dropped off as well. He would hire some people. He would hike around, make observations, collect samples, send those samples home. So he's definitely doing a lot of things similar to what a naturalist would do. The tension between he and the naturalist on board continued throughout this entire 5-year journey. So Darwin's making all of these observations and collecting samples, sending everything back home. We tend to have a misconception about the turnaround time here. No matter where Darwin was, as long as he was at a major port shipping his items back home, those items could get home in about 2 to 3 weeks. The people back home would open up the packages. They would compare them to samples and things that they had in their own history museums and in their own private collections. They would write back to Darwin about how the samples and things were connected and what their thoughts were, and then Darwin would write back to them. So this entire 5 years, Darwin is in continuous communication with his colleagues back home. When Darwin gets home, he steps into the role of naturalist. He does a lot of work with worms and soils. He discovers auxin with his eldest son. Remember, auxin was a plant hormone that causes the plant to grow, produced by the apical meristem. And that movement of auxin from the apical meristem down through the plant towards the roots is what suppresses the axillary buds. As soon as we snip off that growing tip and we remove that auxin, then all of a sudden those axillary buds break dormancy and they start to grow. So auxin is an incredible hormone in plants, and he and his son discovered that. He does a lot of work He's making a lot of contributions to science. He's working on the theory of evolution, but he does not publish for 20 years. This whole time he's in communication with Alfred Wallace. Alfred Wallace is a naturalist. He's making observations in different continents, and he and Darwin are talking about survival of the fittest and the role of natural selection. And Wallace actually had better data to support the theory of evolution and natural selection. So the big question is, if Wallace had better data and there are all these other people who are contributing, why do we only know Darwin? And this comes down to that cliche. Darwin knew the publisher. He knew the editor of a publishing journal. He gets his research in before Wallace does. And here we are. Years later, almost everybody knows about Darwin, and very few people are familiar with Wallace and several of the other colleagues who are working on evolution as well. So, regardless of who we give credit to, this concept of natural selection and survival of the fittest is very helpful for understanding a lot of the diversity of life on this planet. So let's do a quick overview of the evolution of plants, looking specifically for ancestral and derived characteristics. So we know that our planet is very old. The first evidence that we have of life on our planet are these stromatolites. And stromatolites are bacterial mats. So when we're looking here at these individual layers inside of one stromatolite that's been cut in half, what we have to remember is that each layer represents millions of individual bacterial cells, and that each layer is laid down over time, kind of like sedimentary rock. This picture on the right is from California. In California and Australia, we still have stromatolites being formed today. And weirdly enough, the melting of the glaciers in Greenland has revealed a lot of stromatolites. And as weird as that sounds, that has been hugely helpful for our understanding of evolution. So our stromatolites, if you wanted to see one, they've got one at the University of Wisconsin-Madison Geology Museum, and they are about the size of an ottoman. So we've got life starting with these single-celled bacterial cells. And the first life on this planet primarily eats methane and can live in an environment with no oxygen. We didn't have photosynthesis for a very long time. It wasn't until about 2.5 billion years ago that we have the first cells doing photosynthesis. Now, those are algae cells using phycobilins. They're producing oxygen as a byproduct. And the thing that we have to keep in mind is that oxygen caused one of the first major extinctions on this planet. As oxygen levels increased, they poisoned the bacteria that had evolved to eat methane and live in an oxygen-free environment. So we're going to skip really far ahead. We're going to skip to the idea that we have multicellular organisms doing photosynthesis. We're going to skip all the way to the domain Eukarya, where we have cells that have a nucleus and a Golgi apparatus. They have plasma membrane-bound organelles, including the chloroplast organelle, doing photosynthesis the way that we learned about. Carbon dioxide and water and sunlight are the inputs, and carbohydrates and oxygen are the outputs. So let's jump into the domain Eukarya, kingdom plants, and look at those main divisions, including the division of Carophyceans. One of a few, at least, that we know now, a few ancestors to all of the land plants on our planet. So here we're looking at a phylogenetic tree, an abstract tool showing us the relatedness between different species. Down here we have the root connecting to the green algae, connecting to our Carophycean. Our first major group of land plants will be our liverworts. Then we get our mosses. We're going to skip the hornworts and we're going to jump right to our ferns. From our ferns, we get the evolutionary development of all of our gymnosperms, our naked seed plants, and then the plants that we have spent the most time on this semester, our evolutionary event that gives rise to our angiosperms with their flowers and their fruits. I like this phylogenetic tree a little bit better because here we can see that incorporation of the Carophyceans, at least one of 2 or 3 groups of plants that we know gave rise to all plants on land. And we see our liverworts, our mosses, our ferns, our naked seed plants, and then our angiosperms, the flowering and fruiting plants. For each group, each division, I'm going to introduce or show you the alternation of generations. Now, the alternation of generations really is focusing on plants changing from strictly asexual reproduction and also being able to do sexual reproduction. The whole advantage of sexual reproduction is you get to mix up your genes, and maybe the offspring will get some genetic variation that allows them to survive in a changing environment. So just a quick refresher on some of those sexual reproduction terms. Ploid or ploidy refers to the number of pieces of DNA in the nucleus. Diploid is when we have homologous pairs of chromosomes, those two pieces of DNA with genes for the same trait. The prefix di or di means two. Haploid is going to refer to when we have half of each homologous pair of chromosomes. So we just have one piece of DNA for each chromosome position. Triploid is when we have three pieces of DNA for each chromosome position. That prefix tri meaning three. Remember when we saw triploid, this term referred to the endosperm in the seed of angiosperms. Our gametophyte generation, or gametophyte, is going to refer to when we have haploid cells. Sporophyte is going to refer to when we have diploid cells. So with our alternation of generations, we are going to be looking for the alternating of structures, which are either haploid or diploid, gametophyte or sporophyte, in order to do sexual reproduction. Now, I'm not going to expect you to have these memorized, but I do want to introduce you to this because if you go further with botany, they're going to make you memorize all of this, and it gets hugely complicated. We just want an overview of our alternation of generations. So really we want to focus on which structures are haploid. In other words, sexual reproduction is happening primarily with our sperm and egg or gamete cells. Then we want to pay attention to that fertilization event that gives us our diploid, our homologous pair of chromosomes, gives us what potentially could be our sporophyte structure. Now, as we get into each of our division of plants, I'm going to answer three questions. What are the ancestral characteristics that we have for all of the species in this division? What are the derived characteristics? What's new with this whole division of plants? How does that maybe give them an advantage over the previous division of plants? And then I'll give you some other important details. So we have to start with our Carophyceans. And despite the size of this picture, they are fairly small organisms. But some of the ancestral characteristics that we have is we have a plant that is multicellular. And this cross-section of the stem of a Carophycean is showing us that we have multiple cells. So an ancestral characteristic is that we have multicellularity. This plant is doing photosynthesis. This plant is also able to do both sexual and asexual reproduction. So those would be all of our ancestral characteristics. Now we know that Carophyceans have been around for a very long period of time. We have fossilized evidence of Carophyceans. Here we're seeing Carophycean reproduction. We're seeing that asexual reproduction phase on the bottom, and then that important sexual reproduction phase. One of the things that our Carophyceans will have over previous species of algae is they'll have a very clear antheridium. The antheridium, if you go further with biology, does refer to the male structure. If you remember when we learned about flowers, we studied the anther. The anther was the structure that, the whorl that had the male gametes. So let's get into life on land. Our first main land plants are going to be liverworts. And while I adore liverworts, they are fairly pathetic. They're just small little pads of leaf-like cells, but it's not a true leaf. We don't have that tissue differentiation. So an ancestral characteristic that we have with our liverworts is we do have multicellularity, and we can see that in this blown-up picture, all those geometric shapes. we're still going to have sexual and asexual reproduction. This round structure here is something called a gemma cup. This gemma cup is going to do asexual reproduction. So that's an ancestral characteristic. So what are some of another ancestral characteristic will be sexual reproduction. Here we have the Archegonia, and here we have the Antheridia. So we have separate male and female structures. But a derived characteristic for these plants will be that these plants can survive on land. They can tolerate some time without water, but in order to do sexual reproduction, they're still going to have that ancestral characteristic of needing water so that the sperm can swim from the male structure to the female structure, and we can get that change to sexual reproduction. Another ancestral characteristic that we have is that all of the cells that we see here are haploid. In other words, everything that we're seeing in this picture is in the gametophyte stage. Here we're seeing a more detailed view of the alternation of generations for our liverworts, and notice that we will almost never see that sporophyte diploid stage. Most of the time what we are going to see is haploid cells. Our liverworts were around for a while, but seeing as they could never grow very big, they were never going to dominate the planet. Our next evolutionary event is the evolutionary event that gives rise to our mosses. Now again, mosses aren't real big, but they are able to survive on land and capture more resources. So let's first start with those ancestral characteristics. Well, our mosses are going to be able to do photosynthesis. Most of the structure that we're looking at in these three pictures is haploid. So we're looking at mostly gametophyte stage structures. Our bryophytes can do both sexual and asexual reproduction. When they do get around to doing sexual reproduction, they will produce this one sporophyte structure that we see here that looks like a little bulb. With our bryophytes, we are going to have a lot of derived characteristics. So one of the derived characteristics that we'll have is we'll start to have some tissue differentiation. So here we can see some leaf-like structures in these two separate bryophyte species. The cells that are the dermal cells will start secreting a waxy cuticle, so we have that tissue differentiation. Dermal tissue making that waxy cuticle, starting to keep some of the moisture inside of the plant. The other nice thing about a waxy cuticle is it's going to prevent against DNA damage. The other thing that we're going to have is we're going to have small little rhizoids. Those rhizoids, rhizoids, those rhizoids are going to be able to get into the soil a few inches, maybe 2 or 3 inches, get some nutrients and get some water in order for the plant to grow more and capture more resources. So our mosses, our bryophytes, can start to outcompete our liverworts. We have fossilized evidence of mosses as well, so we know that they've been around for quite a while. Turns out that our mosses are part of what eventually, through geological transformation, heat, pressure, and time, our mosses eventually become our fossil fuels, our coal, oil, and natural gas. So here we're looking at that picture of bryophyte alternation of generations. Most of the structure that we're ever going to see will be haploid. Only our little dark structures up here will be in that sporophyte generation, having diploid cells. Our next big division of plants will be our ferns, our pteridophytes or pterophytes, depending on the botanist that you're talking to. When it comes to ancestral characteristics, we're going to have quite a long list. Our pteridophytes are going to have leaf-like structures. They're going to be doing photosynthesis. They'll have a waxy cuticle on their leaf-like structures secreting cutin to help keep the moisture in. The plants will be doing sexual and asexual reproduction. But we're going to have a lot of really interesting derived characteristics. One of the derived characteristics that we're going to have is that most of the structure that we're seeing here is going to be diploid. sporophyte. So we've had a huge shift in the major structure that we see, the major structure that the plant is investing energy in, as far as the alternation of generations is concerned. So we've got that going on. And then the other important derived characteristics. We have rhizomes, those horizontal underground stems that are allowing the plant to capture more resources and spread over a larger area. And we have the differentiation of vascular tissue. We have those tracheid cells. Those tracheid cells are going to allow our ferns to move water longer distances. Why is this important? Well, in a world that's covered with moss, you have to be able to get up above the moss in order to capture sunlight and do photosynthesis. So if you can grow taller, you can capture more sunlight, you can outcompete the mosses down below. So that's why our tracheids become so important. And some of our ferns are able to reach really big heights. So this is a tree fern. They still grow on our planet today in the tropics, even though they've got a lot of gymnosperm trees and angiosperm trees to compete with them now. But they can get as tall as a lot of our normal trees. So it's pretty impressive to see the heights a fern can get to. Most of the time we think of the low-lying ferns that we see here in this picture from the St. Paul Conservatory. If you ever flip a fern over, what you will see are these little brown dots, and those little brown dots will be the spores. Those are essentially the reproductive structures. We have fossilized evidence of a lot of the ferns to see that the fern morphology, those physical characteristics, look very similar to a lot of the ferns that we see today. And we zoom in on the alternation of generations. We see that most of the structure that we see above ground will be sporophyte. Finally, we're seeing mostly diploid cells. We will almost never see this gametophyte structure that grows in the ground. Almost looks like a liverwort. We've got that kind of throwback to previous species that have come before. Now, we almost never see the sexual reproductive phase when the cells are haploid and those gamete cells are combining. Our next big division of plants will be the gymnosperms, our naked seed plants. With our gymnosperms, we still have a lot of ancestral characteristics. They're doing photosynthesis. They can do sexual and asexual reproduction. They've got a waxy cuticle protecting their structures. They can keep water inside. They've got tracheid xylem tissue for moving water long distances. A lot of our gymnosperms still have rhizomes, those horizontal underground stems. So what's the big advantage here for our gymnosperms? What are some of those derived characteristics? Well, in order to understand the derived characteristics, we have to understand the environment on the planet. Up until this point, the environment on the planet had been fairly warm and fairly wet. But when we have a climate shift that makes the environment really dry, that external pressure forces the plants to essentially work to keep that crucial resource, water, so that they can still do photosynthesis. So when we look at the derived characteristics, we have to put it in the context of a drier environment and needing to conserve or preserve water at all costs. So one of the big things that gymnosperms will have is they will mostly have rolled leaves. So by rolling up the leaves, you decrease your surface area. With less surface area, less evaporation. So yay to that. Even the gymnosperms that still have flattened leaves, those leaves are going to be, they're going to be small. They're going to have a very small amount of surface area, keeping more water in. We'll see the development of vessel elements for xylem tissue, a way to move water longer distances and move larger quantities of water so that when it does rain, you can grab lots of it and you can keep it inside of your body. A couple other things that we're going to see for derived characteristics is we are going to see pollen. Remember, pollen is the gamete cell, sperm, inside of a package, and that pollen, if you remember back to the paleoecology lab, can stay intact, can stay preserved for a very long period of time. So you've got your male gamete all wrapped up and protected inside of pollen. and then we look at the female structure. The female structure is going to modify, and we will finally start seeing seeds. We will see that ability for the baby embryo to start to grow inside of the parental tissue for a small period of time, and for that baby plant to go dormant and to remain dormant potentially for hundreds of years, essentially waiting for the right environment. And what do our seeds need to germinate? Temperature and water. Sunlight is not the consideration. So we see within the sexual reproductive structures, we see modifications to cope with the lack of water. Here we're looking at horsetail, or equisetum. And equisetum does grow in our prairies, has the ability to withstand our cold winters, and we see those rolled up leaves, and we see that reproductive structure here up at the top getting ready to make naked seeds. Seeds without any covering, but will be able to protect the dormant plant, give it some food, so that when the conditions are right, that plant can grow again. Remember we talked about ephedrine during the medicinal plant unit. Here we see the ephedra plant growing with those rolled leaves, minimizing moisture loss. The ginkgo tree is one of the trees that does have flattened leaves, but these leaves are very, very thin. They're very small, and if you were to feel them, they have an intensely waxy cuticle protecting them from water loss. More commonly, when we look at the gymnosperms, people bring up pictures of pine trees and the conifers, and these are excellent examples, even if they grow in the mountainous region. You have to remember that in the mountainous region, moisture doesn't stick around, especially at higher elevations with steep inclinations. The water is just going to whoosh through the environment, so it's very much a water-deprived situation. Here we're looking at pine cones, and even though junipers and some other conifers produce this kind of fleshy structure, please understand that these are by no means true fruit. They are not modified ovaries. A lot of times these are arils. They're produced by the seed to help protect the seed. So our gymnosperms have the advantage of making a seed. They're going to be able to withstand drier temperatures. And we're going to see that this derived characteristic gets passed into our group of angiosperms. Again, we have fossilized evidence of equisetum, horsetail, fossilized evidence of ginkgo trees, and we see that our gymnosperms have changed very little since they first evolved on our planet. Looking at that alternation of generation, most of what we see with the gymnosperm is going to be sporophyte. Our gametophyte haploid will only be the egg, ovule, and the pollen or sperm. Now, we have spent a tremendous amount of time this semester talking about our angiosperms. So what kind of ancestral characteristics do our angiosperms have? It's almost a list of everything that came before. So, sexual and asexual reproduction, check. Photosynthesis, check. Tissue differentiation, check. Tracheids and vessel elements in your xylem, checkity check. Cuticle cells with a waxy cuticle to help hold moisture in, checkity check. Seeds, pollen. It's got it all. So what's the big advantage for our angiosperms? Our angiosperms are going to modify their ovary to become a fruit, and they're going to modify some of their leaves to become the flower structure. This will give them more refined control over their sexual reproduction through using pollinators to move the sperm to the ovule, increase sexual reproduction. Now, the interesting thing is when we study the amount or the number of species in each of these divisions, there's not a whole lot of liverworts. There really aren't a whole lot of mosses. There's a few more ferns, but not a whole lot of those. There's not a whole lot of gymnosperms. And then we get to angiosperms. Holy cow, we just get this explosion in diversity. And so the structures and taking advantage of all of the characteristics that have been evolved before, and then developing flowers and fruit, this is what has allowed our angiosperms to really dominate the planet and to give us such diversity. It's one of the reasons why our angiosperms are one of the most commonly seen plants on our planet. So there we are, our review of the diversity of life, kind of an overview of the evolution of plants. So let's come back to Darwin and the theory of evolution. Let's touch on this process of natural selection, because it has so influenced this diversity of life that we see. Members of a population will have heritable variations. In other words, there are genes, chunks of DNA that code for proteins. The population will always make more babies than the offspring, more babies than the environment can support. This means some individuals are going to die. Only those that have the traits that allow them to survive in the environment will be able to survive and pass on their genes to the next generation. So across generations, we see this consistency. We see most of the population has those favorable traits. Natural selection can only be based on genetic possibilities. Evolution is only based on what has come before. And extinction will happen when adaptations are no longer suited to the environment. So when we look at Darwin's conclusions, the theory of evolution has really helped us to understand natural selection being one of the mechanisms for why we get these very incremental changes from one division of plants to the next, and the advantages that those derived characteristics gave to the new division of plants. So we go back and revisit this definition of a species. A species will be members, individual organisms that have a shared gene pool. They're interbreeding and producing viable offspring. Each species is reproductively isolated from other species, and species always share a common ancestor. We're always building on what has come or what has evolved before. So when we have speciation, the splitting of one species into two or more species, we know that we've got changes in the gene pool. Sometimes the physical characteristics are very obvious, but sometimes they're very subtle. The important thing to remember is that once this split has occurred, the species are no longer able to interbreed. This is why you can plant a potato next to a tomato, and while they are closely related to one another, they're not going to make totatos or pomatos. They're not going to make a hybrid tomato-potato. They're just simply not going to be able to interbreed. So this picture of the orchids is showing us that sequential progression, the relatedness between species, but then the idea that when we have this splitting, there are significant differences, even though there are some characteristics that are similar to the species that have come before. I just wanted to touch on the two types of evolution before we wrap up our discussion of plant diversity. The reason why is because across our planet, we see some really important common patterns. So one of the common patterns that we'll see is the result of convergent evolution. So in this picture, we've got euphorbia, and euphorbia are only found in Africa, and we've got cacti, and the cacti are only found in the Americas. The plants look very similar to one another. If I didn't tell you that these are two distinct and separate species found in very different geographic locations, you might have thought that these were the same plant. So why do these two plants look the same? Well, the reason why they look the same is because they are converging on similar solutions to similar problems in the environment. Both of these plants grow in desert-like environments where water is very scarce. So they've developed that modified stem, and then in the stem, we see those individual segments. Now, segmentation is brilliant because when the rain comes, it's going to whoosh, come down quick, and then because of the sandy soils, the water's not going to stick around very long. So when it rains, you've got to grab that water if you're a plant. If you have segments, your segments can swell and swell without splitting. But then over the next period of time, which could be over a year, the Sonoran Desert went 496 days without measurable rainfall. So over the next period of time, you are going to have to use all that stored water, and you're going to contract and contract and contract. Segments, again, are really handy for that because as you contract, you're not going to split to pieces. So segments allow our euphorbia and cacti to expand and contract with the fluctuation of water in the environment. So they're converging on a similar solution. Segmentation to deal with water availability and deprivation in the environment. It's not just those two species of euphorbia and cacti. Here I have another picture of a kind of round, small round cacti and a small round euphorbia. Again, we see the segmentation. We have that modification of the stem and the rolling up of leaves into spines to help defend against predators in the environment. They're converging on similar solutions to similar problems in the environment. We see a lot of convergent evolution with plants. Both the bryophytes and the gymnosperms have these kind of rolled up leaf-like structures to cope with water loss. Here we see a fern and a gymnosperm. The gymnosperm happens to be a cycad. And interestingly, they have similar leaf-like structures. They've got that pinnately compound leaf. They've split that blade into lots of different segments. For the gymnosperm, it was to maintain water, to keep that water from being lost. For the fern, the segmentation, that pinnately compound segmentation, is going to help with water loss, but water loss was never going to be a big issue for the ferns because they lived in a wetter environment. Here we see a gymnosperm, Equisetum, looking a lot like this the fountain grass that we see used in landscaping. So both of these plants have the adaptation of rolling up the leaves to decrease water loss. Remember that our grasses do C4 photosynthesis and are adapted to living in drier environments, even though they are an angiosperm, a fruiting and flowering plant. So we also see divergent evolution. This is when we have very closely related species that look very different from one another. They have very different adaptations, even though they're living in the same geographic location. So the big question is, why would you look so different if everything in the environment is the same? And one of the answers to that question is competition is probably driving that divergence in how our species look, the divergence and speciation. So we've got a lot of examples of this as well. The cycad and the pine tree, I think, are an excellent example. So when we take a look at our pine tree, we've got those characteristic rolled up needles. And then here when we look at our cycad, we can see we've got a blade, but that blade is pinnately compound. But it's a very different way of minimizing water loss than taking your leaf and rolling up tightly into one individual little needle. So divergent evolution happening in our division of gymnosperms between our cycads and our pine trees. Now, humans have definitely been messing with evolution. And the main way that we've been doing this with plants is through artificial selection, selecting for very specific traits. So if we go all the way back to the beginning of the semester, when we talked about the grasses, one of the main traits that we selected for were those non-shattering heads. The parts of the stem that would hold on to the seeds so the seeds wouldn't be spread everywhere, made harvesting easier for us. Through that artificial selection, selecting for that recessive gene, we now have grasses where all the grasses that we grow for agriculture have that recessive trait for a non-shattering head. We look at corn. For corn, not only did we have to select for the non-shattering head, but we also needed corn to have softer seed coats. Throughout all of the plants, we have always been selecting for plants that were sweeter. We have a love of sugar. Artificial selection, as we're seeing here in this slide, with the Brassica family. Remember our mustards. Our mustards, when we talked about the herbs. Mustard family has all of the brassicas, the broccoli, the cauliflower, the Brussels sprouts, the cabbage, the kale. And it's through artificial selection that we get all of this diversity, including the fast plants that you were growing, or hopefully grew, this semester. So through artificial selection, we are messing with plant evolution. We are selecting for traits that otherwise may not persist. I mean, from a plant's point of view, being a seedless watermelon makes no sense. Invest all that energy in the watermelon fruit, but the babies you don't have any babies there? Like, that's weird to put all that energy into making the fruit. Seedless banana, same thing. Why would you invest all the energy in making that fruit if there's no babies there? That's just a lot of wasted energy from the plant's point of view. So we see that humans have really been altering and messing with plant evolution. And this is why having seed banks is so crucial and so important. Remember, our seed banks are where we're holding seeds, preserving that genetic diversity, because our agricultural practices has led to genetic erosion, that irreversible loss of genetic diversity. And we know that sometimes, even in plants that don't make qualities or don't have products that we necessarily enjoy. Take the banana, for example. The wild banana with all those seeds and all those little crunchy pips that are not enjoyable to eat. In those seeds could be the gene that helps us deal with Panama disease, helps us deal with the fungus it's causing the collapse of the Cavendish, that already caused the collapse of the Big Michael, the Gross Michael banana. We know that we have problems on our planet, and that maybe those seeds in the seed bank, in the wild type, have the key to resolving that. And don't forget that when we studied the grasses, our agricultural practices, putting selective pressure on the fungi and the bacteria to evolve and be able to overcome our chemicals, means that we now have UG99, that fungus that we do not have a way to kill, that is attacking our wheat and our grass plants in the Middle East and Africa. These are ugly problems. So we're hoping that maybe somewhere in a wild grass, in a wild wheat, a wild barley, a wild rye, maybe there will be the gene that can help us conquer the UG99 fungus and anything else that comes around. We halt and we alter sexual reproduction. We select for traits that we find desirable. But we have to remember the larger context. The rest of the world is still evolving and still changing. The relationships between plants and the rest of the organisms on this planet generally has not changed. Predators like herbivores are going to want to eat our plants. Predators like bacteria, like fungi, insects, they're going to want to eat and consume our plants. So we have to always work within the relationships that always exist. We have to resolve our problems. We have to practice agriculture in a way that takes the whole context into account. And this is why we learn not only about the individual plants, but we learn about this larger historical context, the evolution of how we got to the plants that we have today, how we got to the situation that we have today, so we can have a better understanding of our biggest impact on the planet, which is what we eat. So, if you have any questions, please email me or drop into Zoom office hours.